Welcome to the influenza simulation. In this simulation, you will use a respiratory cell of a pig, also called a swine, to study how viruses can adapt and move from one species to infect another species that was previously unaffected by the virus. You will use genetic material, called genes, to specify the building of the surface receptor binding proteins associated with the swine influenza virus. Then you will reassort the genes and see how the avian swine influenza viruses combine their genetic material, called RNA. Next, you'll mix swine-human influenza viruses to create novel RNA sequences. Lastly, you'll see how this combination results in viruses that have a different genetic makeup that are capable of infecting new host species. To begin, click on the Sim tab. You will see a swine influenza virus that commonly is found inside a respiratory cell of a pig, aka a swine. Notice that the inside of the virus has 8 strands of RNA. 6 of the strands are black. The black strands indicate that they are non-coding RNA sequences and that they do not make surface proteins. The 2 colored strands code for proteins that are located on the outer surface of the virus. The outer surface of a virus is called the capsid. The surface proteins are critical for allowing the virus to enter the host cell, such as a pig or a human respiratory cell. Each year, scientists create influenza vaccines designed to recognize the surface proteins of viruses expected to infect people. The vaccine protects people from infections by blocking the viruses from entering the human cells. The influenza virus surface proteins come in 2 types, HA and NA. Your first task is to build the surface proteins on the virus capsid. You must assign the NA and the HA surface proteins based on the coding RNA sequences inside the virus. To do this, click and drag a NA protein to the outside of the capsid. The proteins will stick to the capsid. Next, click and drag a HA protein to the capsid. To see if you are correct in your construction of NA and HA proteins, click on the Check Your Build button. If your build was incorrect, click and drag a different NA or HA protein to the capsid. It will automatically replace the existing proteins. You may replace either one or both of the proteins. After you try another combination, click the Check Your Build button. If you are incorrect, continue to try different combinations until you have the correct combination. Once you have the correct combination, click on the Reassort Genes button. On the next screen, you will see an influenza virus found in a pig cell on the upper left, and an influenza virus that lives in a duck, aka avian, cell on the right. Clicking on the avian swine button will make the RNA inside a swine influenza virus undergo reassortment with an avian virus. Next, click on the swine human button. You must finish building the surface proteins to make the swine human virus. To do so, click and drag the NA and HA proteins to the capsid as you did on the previous screen. Click the Check Build button. Continue to try different combinations until you have the correct combination. Once you have the correct combination, click the View History button. This will show all the influenza's RNA sequences that you created for the pig, duck, and human hosts. You can now click the Reset button to clear the screen and go back. Click on New Virus to begin with a different virus.